Hello, my name is Allison Mool, and I had the privilege of working in Dr. Nee's lab this summer. I am also about to be a senior at Illinois State University, and I am currently applying to medical school. And so when I found out about this internship and the opportunity it would bring to do research that could directly affect patients, I was very excited to get started. So my project this summer was called A Role of TMEM97 in Tumor Cell Autophagy in Responses to Metabolic Stresses. So when I first entered this research lab, I didn't know a ton about cancer research techniques. So the first thing I learned how to do was culture tumor cells. And this really is the foundation of cancer cell research because if you don't have cells alive and able to be experimented on, then you can't do experiments. And so the next thing I learned how to do was a migration assay. And so this is when you can add a treatment or manipulate a variable and see how this affects the cell's ability to migrate. I also learned how to do viability measurements, and this is a similar process to the migration assay where you can manipulate a variable or add a treatment and see how this affects the cell's survival. I also learned how to do immunocytochemistry and a Western blot, and we'll talk a little bit more about those two techniques later. So before we get too far into this presentation, I wanted to talk to you a little bit about the cells we used this summer. So we used an MCF7 cell line, and this is breast cancer tumor cells. We also use LINCAP cells, which are prostate cancer cells. And Dr. Nee's lab is very interesting because they're looking at the sigma-2 receptor, also called a TMEM97 protein. And so his lab has engineered a cell line that overexpresses the sigma-2 receptor. And they also have a normal cell line of normal sigma-2 receptor expression called PCDH. So if you say that you are looking at cells that are MCF7 TMEM97, you're looking at breast cancer cells with this sigma-2 receptor overexpressed. If you say you're looking at MCF7 PCDH cells, you're looking at breast cancer cells with normal sigma-2 receptor expression. And so we're looking at the sigma-2 receptor for a couple reasons. One, we don't know much about it. Frankly, in the grand scheme of research, it's a somewhat new finding, but we do know that it's associated with cancer drug resistance, and it's associated with the autophagy process. And so what is autophagy? Our lab specifically looks at chaperone-mediated autophagy, and this is a sort of survival response. So when a cell is in stress due to nutrient deprivation, or if it's in the body and we're targeting it with chemotherapies and hormone therapies to try to stress this cell and kill this cell, it can use its proteins and organelles and sort of eat itself to make energy and survive in these stressful circumstances. So if we can somehow manipulate how well this autophagy response works, then we can affect how well our treatments work and how well we can kill these cancer cells, which is ultimately the goal. So we know that LAMP2 protein is in the autophagy process. It is on the lysosome membrane, and so when something is targeted for degradation, it kind of guides it into the lysosome, and then enzymes can degrade it inside of this lysosome. And so we know LAMP2 is involved in autophagy, and we know for the most part how. The sigma-2 receptor we know is associated with autophagy, but we don't understand the mechanism yet of how exactly this works. So our lab wanted to see if the sigma-2 receptor somehow interacts with the LAMP2 protein specifically. So the first experiment we did when looking at this research question was to plate out MCF7 cells, so that's those breast cancer cells, and then we treated them with rimcazole. So in our lab we call this a rim treatment. And this rim treatment is a sigma-2 antagonist. So this means it prevents this sigma-2 receptor from working to its full capacity, or it inhibits the sigma-2 receptor. And then we put this in a stressful environment, so this autophagy process starts working as the survival strategy. And then we can visualize under a microscope how treating these cells with RIM, so inhibiting the sigma-2 receptor, affects the distribution of this LAMP2 protein. And we use antibodies to visualize this. And in the next slide, you'll see this red fluorescence that represents the LAMP2 protein. So here are results from this experiment, and we did see a correlation in manipulating the sigma-2 receptor function and altering the distribution of the LAMP2 protein. So in these images, you can see this blue color are the nuclei, and then this red color is the LAMP2 protein we're looking at. So on the left here, you see a control. So this is when the sigma-2 receptor is working at normal capacity. And you see a sort of punctuated or speckled appearance to this red color. So the LAMP2 protein has a speckled appearance on the left. And then on the right, when we're treating this sample with RIM, so when the sigma-2 receptor is inhibited and not working as well, you see more of a solid appearance of this LAMP2 protein or uniform distribution. And so in these samples, we also saw a similar distribution pattern. So our lab wants to know, is this just a difference in distribution pattern, or is this a difference in quantity? Are there different amounts of LAMP2 protein in one of these samples? And so moving forward to find out more, we decided to use a Western blot. And so we treated some samples with RIM once again to inhibit the sigma-2 receptor, but we also used a PB28 treatment. And this is a sigma-2 agonist, which means that it activates the sigma-2 receptor and therefore increases the function of the sigma-2 receptor. So when we use these opposing treatments and cause opposite effects on the sigma-2 receptor, if it does directly affect the quantity of LAMP2 protein, we should see the opposite effects on the quantity of the LAMP2 protein between these two different treatments. So this slide is an overview of the steps of a Western blot, and you can pause here and read if you'd like. But basically, the Western blot allows us to separate these proteins and then use an antibody to detect the target protein we're looking for. In this case, that's LAMP2. So here are our results from the Western blot. If you look on the right side of the figure, you can see the two leftmost columns are labeled control. So this is when the sigma-2 receptor is working at its normal capacity. And you can see the LAMP2 protein we detected in line with the arrow. And if you move to the right, the next two columns are the PB28 treated cells. So this is when the sigma-2 receptor is working at higher capacity than normal, and you see less LAMP2 protein than in the controls. And if you move to the last two columns, you'll see the RIM treated cells. So this is when the sigma-2 receptor is inhibited, not working as well as normal, and you see more LAMP2 than the controls. So we do see this opposite shift that we were kind of expecting from these two opposite treatments. And so we can see from these results that in some way the sigma-2 receptor is affecting the quantity of the LAMP2 protein. So what's next? What does this mean? Well, we don't quite know yet. That's what's kind of exciting about research. We found these breadcrumbs, and now Dr. Nee's lab gets to follow these and discover more about what's happening on the cellular level and hopefully put the pieces of this mechanism together. So perhaps one day in the future, we can use this sigma-2 receptor as a target to affect how well this autophagy process and response works in cells. Down the line, maybe we can use the sigma-2 receptor as a target and affect how well our cancer cell drugs work, which is a very exciting prospect and could be very, very exciting in the field of cancer research. I'd just like to say a quick thank you to Dr. Nee and Sheng Wei and Dr. Wang for welcoming me into their lab and taking the time and energy to teach me this summer. I've truly enjoyed working alongside you and all that I've learned this summer. I'd also like to thank anyone who makes this internship possible and supports cancer research. And thank you once again for listening to my presentation.